Hi everyone, I'm Rachel Hostetler, and today I'm joined by Chris Morrison and Scott Daniel from the Allen Institute. Today we'll be showing how to programmatically access data from the Allen Brain Cell Atlas. This webinar will contain 5 parts, so feel free to skip any if they're not useful for you. The first part is the installation guide, followed by the data model. Then we'll go over cell type taxonomies. Next, working with H5AD files. And finally, a use case example. At the end, we will also share helpful resources for using these data. All right, starting with our installation guide, I'm just going to go over a few quick and easy ways to get the computer ready. First, I'm going to go to anaconda.org and download Anaconda. And Anaconda is an open source library management system, and it's used for creating and managing virtual environments. And there's many other options to use besides Anaconda. This is just the option I personally like. And here you can see different options for more information about what's in Anaconda, what the accounts are, and if you already have an account, you can use that, or you can click on Get Started. Then at this step, you can verify your email. Once you've verified your email, it's time to choose what type of machine you have. So I am currently using a Windows machine, so that's what I'll be using for the rest of this setup. But if you have Mac or you use Linux, please use those options. Next, we get to the installation wizard, and there's a few options we have to set up. We can click on the installation type, we can select the location, and we have some advanced installation options as well. Once you get through all of these, we can then start to install Anaconda. And this will take a while, so you have a couple minutes to go do something else. While we're waiting for Anaconda to download, we can go back to our browser and search for GitHub Desktop. Then we can go to the Download GitHub Desktop website. And again, I'm using Windows, but there are other options if you're running Mac. But for me, I'm going to click on Download for Windows 64-bit. After the download, we'll want to open GitHub Desktop and first clone a repository. And you can clone a repository from your GitHub account, but we're actually going to be using a URL. And we're going to be cloning from Allen Institute /ABC_Atlas_access. And you want to make sure that you have a new folder that you're cloning into. Our next step is back in Anaconda. So you'll want to launch Anaconda. Then you'll want to go to Environments. Then in the Environments tab, you'll want to click Create, and then create a new environment called ABC Atlas Access. Afterwards, you'll want to check the Python box since this involves Python packages. It'll take a few minutes, but once you finish creating your environment, you'll want to go and look at the uninstalled packages, and then we're going to search for Git, G-I-T. Then we're going to select Git, and then we are going to click on Apply. After we have Git installed, we want to make sure we're still in the ABC Atlas Access environment. Then we'll go back to Home, and now it's time to install Jupyter Notebook. After you've finished installing Jupyter Notebook, you can then click on Launch, and it'll launch in the browser. Remember when we cloned the ABC Atlas Access repository on our GitHub desktop? Now we can access that here through Jupyter Notebook. So we can go down, click on Notebooks, and we can see all of these different notebooks containing different types of data sets and tutorials. But before we use any of them, we first want to run through the Getting Started notebook. Again, you need to run through the Getting Started notebook first. In addition, we also have a repository specific to this webinar. So under Allen Institute /SPOCT, SPOCTT, you'll find a directory called 2026_02_11. And this will contain all of the tutorials today that you'll see in this webinar if you'd like to follow along. If you'd like to clone the webinar repository into your GitHub desktop, you can do that as well. Just highlight that URL, go back to GitHub desktop and enter that under clone a repository. Make sure it's in a new folder and then you're good to go. Just a reminder that there are many other resources and installation guides out there. If you search on YouTube, look for Anaconda, Python, Jupyter Notebook, Spider. And again, I personally used Anaconda and GitHub Desktop because that was my own personal preference, but you can look up other installation guides if you'd like more guides on Mac. Here are some examples from a YouTube channel called Programmer Knowledge. Now we'll be joined by Chris Morrison, a software engineer at the Allen Institute. He will be going over the data model used in the Allen Brain Cell Atlas. The notebook that Chris is using can be found on the Allen Institute repository, specifically under SPOCT, SPOCTT, and in the directory 2026-0211. The notebook is titled ABC Atlas Access Tutorial.
All right, for this part of the webinar, we're going to be going over the actual data model and how to find data and where you can find it. So the main place you'll want to start from is this Allen Brain Cell Atlas Access data access page, aka ABC Atlas Access. This is a web portal where we're going to list all the data, all the tutorials that will walk you through how to merge, how to use that data will live. If there's anything you miss or anything I go over too quickly on this notebook, note that this is available for you guys to look at and to use. So all of this will be available. So just going over the high level things. So on that main page, let's go back a second. First off, I'll just say what I'm going to be going through. So I'm going to talk about what data we actually have available on that page currently. Note that in the future, if you're watching this, there will probably be more available. And after that, we'll go dig deeper into a specific projects of what those data are, like what they look like, what you can expect to find. Finally, then we'll introduce how to download the data and how to interact with it, namely through pandas tables. And hopefully by the end of this, we'll make a plot and we'll get to look at things. So very high level, there are effectively 7 data sets that are currently available. They can largely be broken up or conceptually broken up into sets of mouse data. So there's 4 of those, 2 whole mouse brain data sets that one of which includes both spatial and single cell data, and one that is a combination of the whole mouse brain data set here with an external one from the McCosker lab, a set of Merfish data that is also mapped to the taxonomy from this whole mouse brain data set, an aging data set, so you can look at adult versus aged mice with its own taxonomy. There's human data with a taxonomy in roughly 3 million cells from the whole human brain. There is data from the Aligning Science Across Parkinson's project that is mapped to the whole mouse brain data here, taxonomy, and the CAD taxonomy. And finally, there's a multi-species data from the HMBA basal ganglia data set that is human, macaque, and marmoset with its own taxonomy. So there's basically 7 data sets that you'll find. In terms of structure, you can largely think about it as a project, which is one of these 7 things here, or at least currently 7. Directories within those, so these are different modalities. So are they single cell transcriptomic data from 10x? Are they spatial transcriptomic data? Are they the taxonomy folder? Are they the 10x data but for this species, or the spatial data but from this species? So largely these are broken up together. We'll see the actual examples and names of these in a second. Largely, each one of those is going to have data files that contain the gene expression. These are an H5AD these are AND data files. Metadata is just in simple ASCII CSV files. Image volumes, which we're not going to get into here, but those are nifty, and we have some GeoJSON polygon files that we can plot over things. There's tutorials for using all of this stuff. And then finally, a map my cells directory, which has the models that map my cells uses. um, derived from those taxonomies. We're not going to get into that very much, but these are largely all the kinds of data you'll find. Now we're going to drill down a little bit more, go to a specific example. So we're going to use the human mammalian brain basal ganglia data as our example here. So from this project here, there are largely three directories, largely three sets of data that you'll find: the single cell, the spatial transcriptomic, and the taxonomy. Again, those are sort of broken up by the individual species expression matrices and a line data set. For the single cell data, taxonomy, and three different donors from three different species for spatial data sets. So these names here are directories that you'll see later on in this tutorial when we get into what our cache object that will help folks download and interact with the data. So let's drill down a little bit deeper. So we're going to look at specifically the HMBA 10x multiome data. So this is both the expression, the metadata there, but we're also going to look at the taxonomy that is derived from these single cell transcriptomics. So what you'll find in this data set specifically, but also in a large majority of them, is two different files at least. So for you'll find like H5ADs that have raw counts and H5ADs that are normalized to either a CPM or CPT log2 data. These are very lightweight, quote unquote, H5ADs, as in they have a very stripped down obs and a stripped down var. All of that information is basically in the metadata. So if you're usually used to working with large monolithic H5AD files, your obs and var are sort of split out over all of the other metadata that we provide to make these H5AD files not maybe slightly less large than they normally would be if we have all the metadata just loaded into these. Speaking of the metadata, again, looking at this 10x BG inline data. So there's a cell metadata table, a donor metadata table, a library table, a gene. Those are probably the primary main four you'll see in almost every data set, at least for 10x. You'll see donor. For the spatial data, you might see specimen or some other additional tables. All of these data and directories will have tutorials that will help you walk through what data is available and how to join them together to get useful science out. In this case, for this data set, we're just going to be looking at these four metadata tables primarily. These are all keyed and have foreign keys as well. So the cell metadata table has cell label, and that contains a donor label and library label, which you can use, if you're used to database terminology, use to join with a donor, which is going to have your donor ages and sexes and so on. So this is where all of your useful metadata about what a given cell, where it comes from, what its properties are, will live. Another important thing here is the value sets, which have all the colors and ontological plotting orders for, say, the region of interest or the donor sex. For the taxonomy, there's a large number of files that define our taxonomy. I'm not going to get these too deeply. We're going to have a tutorial there in a second, but you'll normally find a cluster, which is a leaf, all the leaf nodes, cluster annotation term, which basically defines what the structure of the taxonomy is, how many levels it has, who's connected to what. The cluster annotation term, which is a definition of all the individual taxons, including their parent-child. Cluster annotation membership, which is sort of more complicated mapping of the leaf taxon to every single level, every parent's level, including itself. This makes for like count statistics of like how many clusters are there in a given subclass, for instance. Easier to do with pandas group bys, which we'll get into a little bit. And then finally, you'll find your cell to cluster membership. So this is how to map cells into the taxonomy. And then the UMAP itself. So these 2D embedding coordinates. So continuing on, these links here that you'll see at the top of each one. So these are, again, the directory names. These link you to the page, and that web page in ABC Atlas Access has links to the data on S3, and you'll find all these files. So that is one way you could do it, but one thing we do provide that makes this the programmatic access tool is we provide the ABC Atlas cache. Now, this is a lightweight object in Python, a lightweight library, effectively, that once you give it a directory on your disk that you want to write data to, it will, once you select, say, I want to download this cell metadata from this project, if you don't already have that downloaded, it will download it and then serve it up to you as a pandas data frame, um, uh, or just the raw path to where is this on disk if you want to just use something else. So we're going to get into effectively how to start using this access cache. So the first thing to do that we're going to do, so this I'm going to assume a little bit that you don't have too much experience using Python. Apologies if you do, and this is all just boilerplate for you. So normally this is what it looks like. So we're going to import the library pandas. This is a way to interact with tabular data in Python. It allows for a bunch of statistics functions, and then slicing and dicing, joining of tables together, and eventually we're going to use it to well, we're not going to use it to plot. It's not going to be the thing that plots things, but we're going to use it as the functionality to allow us to plot. Uh, we're going to talk too much about pathlib. This is basically just a thing to point you at where your file system is. Finally, the thing we are actually going to be using here is the ABC project cache, which will import from the library here. Rachel should have talked about previously about how to install this package, which it is in this environment. So if we just run this bit of code, we'll start this import and get these data together. So what I'm going to do is I'm going to specify where I want to put the data. So that's this line right here. I'm going to set this download base variable. This is basically just a relative location. So we're going 2 levels up in my directory structure, creating a folder called data and creating a folder called ABC Atlas if it doesn't already exist. You can change this to wherever you want on your system. After that, we're going to put it in, we're going to instantiate our ABC project cache using this from cache_dir, which is basically just a catch-all method for creating the ABC cache object we're going to be using. So once we do that, we're going to ask what our manifest is that we're using. And the manifest that we have is the 2025-1031. So this manifest, this JSON file, is basically telling the cache object where all the data is on the cloud, and also keeping track of what data you've already downloaded so that you don't have to keep re-downloading, say, a 70 gigabyte H5AD file over and over. You can download it once and then just load it. So now we have this cache instantiated. If we list the directories, so this is basically every single folder that contains files on the cloud. Here we can see our one that we're going to be using. So in this example, I'm just going to be looking at the 10x multiome BG data and looking at the taxonomy. You'll also notice all of these other HMBA datas. Here's some whole mouse brain. Here's some spatial transcriptomic data. If you want to know more about all of these data sets, the landing page at the start of this tutorial is where you want to go to learn what is in each of these folders and what data set, what project it joins up to. There are also tutorials that will walk you through how to use and line these together on that same landing page. Okay, so then we have all those listed. Let's start listing individual files. So again, mentioned before in this tutorial, so we have our expression matrices we can look at. So that's what this method right here, we input the folder we want, and it lists the raw and the log2 normalized file that we can download. Same with the metadata files. We see cell metadata, donor, library, gene, as described above. And then if we list the taxonomy files, again, we can see all the files that I alluded to above in this tutorial. So this is the actual meat and potatoes of how this is structured. If you ever forget a functionality that's in the ABC cache object and you want to remember, there's a helpful little function in Python called dir. I use it all the time to figure out like what can I actually do. So here's all the, list expression matrix files, list directories. What's the latest manifest file I can download? Um, can I get the file path to any file? Can I download entire directories? Yes, you can. Can I see how big that directory is going to be before I download it? Absolutely. So you're more than welcome to play out this on your own. So let's get into learning the data. Quick aside, there's a helpful little function where you can put at least in Jupyter and IPython, if you put a question mark in front of something, if you have a method you want to know how do I use it, you can put a question and it will list you the things you need to put in. So in this case, I need to put in a directory and the file I actually want to interact with. So we're going to be using this get metadata data frame to load our cell metadata table. This is going to be your main function for interacting with the metadata table, period. And it returns a pandas data frame. So let's do that. Let's load the cell metadata table. So it's going to take a second. It's a fairly large file, but it's complete right now. So you can see all of those columns that we've mentioned above, the cell met label, which is our primary key for this table, donor label, which helps us associate this cell to where the donor came from, the library to the library table, and a few other quality control metrics and IDs that are useful in various contexts. You'll notice this table is currently just indexed by account 0, 1, 2, 3, 4, but that's not quite as useful as we need it to be. So let us set the primary key as the index. This in pandas allows you to look up things quickly. So now this tabular data is indexed by the cell label. So we can just reference these really quick to look up things rather than searching for these specific numbers. If you aren't familiar with pandas, we'll go through some of it here, but here's the link to the pandas data frame documentation. And I believe at the top I mentioned, yes, there's just a link to the pandas landing page. So everything you need to learn about pandas will be on those pages. All right. So now that we have our table loaded and our table indexed by cell label, so our primary key. Oops, let's not do that. If we have a key that we say want to reference, we can do so rather quickly. So this is just accessing that. So cell metadata.loc grabs by the index of the table. So in this case, we have the cell label here, and this is the data that's there. You don't have to do just one. So if we feed it a list of values, we can grab 2 rows, and you can extend this to any n row length. We can even grab the same one twice, I think. Yes, we can even grab the same one twice. And now this is a pandas data frame with a duplicate index. So if we want to grab individual columns, you can do that in pandas by in between the brackets, just specifying no loc here. The column you want to load. Head here is just printing out the first 5 rows. So that's how you access columns. You can access multiple columns, much the same way we did multiple indices. So here we're just grabbing library label and debit score. And then finally, you can combine the two. So we have the indices that we want to grab and the columns that we want to grab. So right now we've pulled a subset of the frame that has 2 rows for our 2 different cell labels, and then the values from these 2 columns. This is the only way you can interact with this data. With tabular data in pandas, you can also slice. There's a ton of other accessors. I would recommend going to look at this. But some useful concepts in Python, for instance, are slicing. So this example here is grabbing the first 100 rows. This one is grabbing a 100 rows from somewhere in the middle that I specified as 1, 2, 3, 4, 5. This is grabbing the last 100 rows. This is grabbing every 10th row. So if I run this table that is produced is every 10th cell. One of probably the most useful ways to interact with this data is through masking. Let's say, as an example, that I have a specific donor that I want to look at that is a donor label in this cell metadata table. So if I want to only look at cells that are in here, I can create a true-false mask. So this is basically for every cell label. It says, is the value of donor label this value or not? So we see a bunch of trues and then false. And then if I use this again in the square brackets, I can grab only data that are from this donor. If I actually do this, you can see that it is much, much smaller than, say, the 3 million cells we usually have. I'm not going to get too deeply into this one, but one of the other very useful things in pandas is our group by functionalities. So let's say I wanted to get the count of cells for every donor that are in this table. So we can do that by creating a group by on the donor label. And then we specify some column here. I just picked cell barcode. But we can ask for the count. And this produces the count. So for every donor, we have the number of cells that are in that donor. Another important thing, so we're going to load the donor table is joining. Another important thing concept is joining data frames. So here we are going to take our cell metadata table and we're going to add information from the donor table into it. So the first thing we're going to need to do is load the donor table. If you've done so here's the donor labels, the species it comes from, its scientific name, the sort of genus, so the common name, what the donor sex is, the donor age, a value for the donor, the units, and then this is actually a hash you can use to look up the donor on NIMP, at least in this example. So let's join this into the cell metadata table and create a new table called cell extended. So we have our cell metadata table here, our donor label, and we're going to use the join functionality and we're going to join it on donor label. So if we hit go, this is now joining the two tables. So we can see our cell metadata table we had before, but now it has all the donor information in it. So this is very useful. Now we have all cells with all of their donor information. Right, here's another example of a join here, which is we can load the cell coordinates. So these are going to be the UMAP coordinates. We're going to load them from the taxonomy directory with, and the file we're going to load is the cell2D embedding coordinates as listed above. We're going to set the index as cell label. So now our cell extended table will have the same index, or the cell coordinates will have the same index as the cell extended / cell metadata table. And we don't have to specify the on here for the function. So if we just run this will join the table. So now we have UMAP XY. So let's plot up some data. So the first thing we're going to do is from our align directory, we're going to load the value sets and then set the index on label. So you can see here we do have species genus in this table. So human macaque marmoset. It has a color, a order that we can plot it in, and some external identifiers and information. So the cell here we're going to run is going to, for the value sets, that's where the field is specifically this. We're going to just change our name really quick to a thing we can index into the cell metadata table. And we're just going to call the new table that we have species color. So we have the label, what genus it is, And we have the same information basically as above. We just renamed one, the description to species genus. So then we have this, we're going to join it on. So we're going to use this table. We're going to grab only the column color hex triplet, which we can do here. And then we're going to say we're going to join it on species genus, which is a column in our cell extended, because we've joined it with the donor table. And let me just plot this out or show this up. So now just showing the columns for X, Y, species, genus, and color hex triplet. So we can see all the cells, their X coordinate unit, their Y coordinate unit, what species they are, and then the color associated with that. Finally, we're going to just use matplotlib to do a quick plot of these. So we're going to import matplotlib. This is just a Python library. We're not going to get too deep into it. If you want deeper examples of this, again, go to the ABC Atlas Access webpage and look at the tutorial notebooks. There's a lot deeper tutorials on how you can plot and visualize these data. So we're just going to import this package, create a figure with a size of 10 by 10, and then we're going to create a scatter plot for every, again, here using the slicing, we're going to use plot every 10th object because we want to plot this decently quick for this tutorial. We're going to grab the X, we're going to have the Y, and we're going to color it by, that's what the C equals, the color hex triplet, set a size, and then give it a marker style. And we should have a UMAP plot, or replot a UMAP plot for the species. If you want a version of this that tells you which color is which, I'll let you in the right corner. Again, the tutorial is ABC Atlas Access for the 10x clustering. For one of the HMBA BG tutorials, we'll have a very pretty plot that will have a legend telling you everything you need to know. Yeah, and at the end here, there's just a link to that specific tutorial I mentioned, and a more direct link to all the information you're going to need at the ABC Atlas Access webpage. So after this, I'll just hand it over to my other colleagues.
Now we'll be joined by Scott Daniel, also a software engineer at the Allen Institute, and he'll be going over the different cell type taxonomies available. The notebook that Scott will be using is in the same directory as the previous one, and it is titled Cell Type Taxonomy Model.
Hello, my name is Scott Daniel. I'm a software engineer and data scientist here at the Allen Institute, and I'm going to be talking to you about how we represent our cell type taxonomies in the data and metadata served through ABC Atlas Access. I will be walking you through this Jupyter notebook that gives some examples of how to download and manipulate one of our cell type taxonomies. This notebook is available in the repository that Rachel mentioned earlier. It's going to be in a slightly different form. I've stripped out most of the extraneous text for the purposes of this talk because I know it can be annoying to read a huge block of text while someone is talking at you, but the code I am executing here is all available in the notebooks in the repository that Rachel mentioned. So let's get going. Let's start by creating our ABC Atlas Access cache object. And now we're ready to go. There are 4 taxonomies available through ABC Atlas Access at the moment. The cross-species HMBA basal ganglia taxonomy, the whole mouse brain taxonomy from Yao et al. 2023, the whole human brain taxonomy from Saletti et al. 2023, and a human medial temporal gyrus taxonomy from our Seattle Alzheimer's Disease Consortium. These taxonomies are all more or less represented the same way through a series of CSV tables that serve as normalized, like they function like normalized tables in a database. And so what we're going to be doing is downloading those CSV files and linking them together to create useful pandas data frames to help us navigate the cell type taxonomy. Before we do that, though, let's talk about how the tables are organized. Keeping the database analogy, there are 2 important columns when talking about a cell type taxonomy in ABC Atlas Access. There is the idea of a cluster annotation term. These are the actual taxons in the taxonomy. These are the biologically interesting groups into which cells have been assigned. And then there are cluster annotation term sets, which are groups of cluster annotation terms. For those of you familiar with our taxonomies already, these term sets are the equivalent of like class and subclass and supertype in the whole mouse brain taxonomy. We refer to those terms in several ways. First, every term and term set has a label. This is a stable identifier used to link the terms and term sets across the tables in the simulated database. These labels are not biologically informative. They are technically human readable, but they're really meant to be read by machines. Every entity also has a name. This is a human readable identifier that has some of the interesting biological information that our scientists have identified, whether the annotation term is associated with a specific anatomical dissection or a specific gene. That will all be encoded in the name. Finally, there's a notion of a cluster alias, which is another identifier for cluster annotation terms that is meant to be machine readable. These are identifiers that refer specifically to the computational clusters that come out of whatever algorithm was used to derive the cell type taxonomy originally from single cell data. Often these cluster aliases do not refer to anything that is necessarily biologically meaningful. However, those computational clusters that have cluster aliases are grouped by our scientists into the biologically interesting cluster annotation terms. The cluster alias is how we link the cluster annotation term to an individual cell in our data set, as we will see later. These columns are all defined in a series of CSV files, and these are the important ones to keep track of when working with the taxonomy. There is the cluster annotation term set file, which just defines all of the cluster annotation term sets in the taxonomy. Remember, those are the levels or the groupings of taxons in the taxonomy. There's the cluster annotation term file, which defines all of the individual cluster annotation terms, all of the taxons at every level of the taxonomy. This file is also notable because it will contain any parent-child relationships between taxons, which is useful for constructing the taxonomy as a tree. There's the cluster-to-cluster annotation membership file, and this is where we link those cluster aliases, the identifiers of the computationally data-driven clusters, to the taxons, to the cluster annotation terms in the taxonomy. And finally, cell-to-cluster membership, which links cells to cluster aliases. So you would go cell to cluster alias to cluster annotation term to cluster annotation term set if you're trying to link everything together in a giant chain. Okay, so that's kind of the setup. That's the background. Let's actually work with the taxonomy. We're going to work with the HMBA basal ganglia taxonomy just because we are. It's the newest one. So that's the directory for it. I will first list all of the files that are available with that taxonomy. And you can see, as I said, there is the cluster annotation term file, cluster annotation term set, cluster to cluster annotation membership, and cell to cluster membership. There are other files in that taxonomy directory, and you are welcome to explore them and see what they contain. But those are the four that are necessary to really construct the structure of a taxonomy, which is the goal of this talk. So let's start with the first file, cluster annotation term set. I'm starting there because it's the smallest. Remember, this is just the one that lists all of the cluster annotation term sets in the taxonomy. You can use the ABC Atlas cache object to download the file and get the path to it. I downloaded it before I came here, which is why you don't see any downloading happening, but that's where it's stored on my local system. I can also just load it as a pandas data frame using this get metadata data frame function. Boop. And then there. That is the data frame that we have. Again, it just lists the cluster annotation term sets in our taxonomy. In this case, there are neighborhoods, and there are classes, and subclasses, and groups, and clusters. But as promised, every term set has a label, which is this technically human readable, but really kind of machine-friendly stable identifier, and then a name, which is much more human understandable. This doesn't say anything about the parent-child relationships between these term sets. To do that, you have to look at the cluster annotation term file, which is what we're going to do now. So we can load that as such. We now have a data frame. Just to see what it looks like. Here there are 1,500 rows. This is one row for every cluster annotation term in the taxonomy. Every taxon at every level has a row here. And again, as promised, they have labels and they have names. They're also associated with cluster annotation term sets. So here are labels. This column here tells you which term set a given taxon belongs to. So these are all neighborhoods, as you can see from this column here. And these are all what are called clusters. For convenience, we also have the name of the term set. If they have a canonical color associated with them in the visualizations from the paper, those are listed here. Useful ordering of the terms is listed here. And then also those parent-child relationships. Every row that has a parent, the parent gets referred to here in parent term label and parent term set label. That tells you what taxon or what cluster annotation term and what cluster annotation term set the parent of this cluster annotation term belongs to. And so that's how you can reconstruct the parent-child relationships amongst all of the taxons in the taxonomy. So we now have enough information to actually construct the entire taxonomy in a single data frame. So let's just do that. Remember, I'm just going to reprint cluster annotation term set here for convenience. So before we construct the entire taxonomy, let's just do some simple proofs of concept. Let's say we wanted to create a data frame that only had subclasses in it. We see from the cluster annotation term set table that subclass is referred to here as CCN 2025-0428 level 2. So we can take the cluster annotation term data frame and subselect it so we only have the rows where cluster annotation term set label is CCN 2025-0428 level 2. We do that. And now our 1500 row cluster annotation term set data frame has been pared down to a 51 row data frame that only contains the subclasses. Now, similarly, let's say we only wanted the classes. We can see here, they all have a unique cluster annotation term set label. They're all just level 2. Now, what if we only wanted the classes? We'll do the same thing and only select CCN 2025-0428 level 1, which was the label associated with classes. We do that, and now we see we have a 15-row data frame that only has the classes in it. And we can just look at the, cluster annotation term set label is uniquely level 1 everywhere. Now we have subclasses and we have classes, and they have a parent-child relationship. I know that because I've done this already. And so we can join the two data frames on that parent-child relationship. So let's just do some bookkeeping first. We're going to just pare down those data frames so that we only have the columns we want, namely label, name, and parent term label. So now I can just show my just subclasses. That's all I got. Label, name, parent term label. Ditto for, we did the same for the classes. And now we're going to rename the columns in our data frame because it's going to be a little confusing because both subclasses and classes just have these three columns. They have a label column, a name column, and a parent term label column. But if we try to combine them, we're going to want to tell the difference between a class label and a subclass label. So we can use Pandas functionality to rename the columns of our data frame. So we're going to rename label, in the just classes data frame, we're going to rename label to class label, name to class name, and parent term label to neighborhood label, because we know that the neighborhoods are the parents of the classes. Similarly, in just subclasses, we're going to say rename label to subclass label, name to subclass name, and parent term label to class label, because we know that the classes are the parents of the subclasses. So we do that renaming. And now if we just look at just classes, we see, there we go, class label, class name, neighborhood label. And then if we look at just subclasses, similarly, subclass label, subclass name, class label. So now we can actually join these data frames. So we have two data frames. We have just subclasses and just classes, and they both have a class label column. And so that's what we will join on. We will take our just subclasses data frame, and we will join it to just classes on class label. We have to set the index of classes to class label to make that join happen. But here I will join that. And now I have subclasses and classes, which is a 51-row data frame. Remember, there were 51 subclasses. And you see we have subclass label and subclass name, class label and class name, and neighborhood label. We've now joined the taxonomy from subclass up through class up to neighborhood. And so that's how we can construct these parent-child relationships using these data frames. So now let's do the whole thing. I'm just going to create a lookup table to quickly map between labels and cluster term set names. And so now I'm just going to write a quick function, or a quick block of code, I guess I should say, that will create a giant data frame with every level of the taxonomy in it. We're going to loop over the levels, starting from the smallest, the child the child that has no children itself, and then up through the various parent levels of the taxonomy. And at each level, we will grab the cluster annotation terms that correspond to that level. And then we will remap the names of the columns in that subsetted data frame. And then we will join that data frame to our HMBA taxonomy data frame, which is the final product we're producing here. So let me just run this block of code. Again, it's available in Git. You can look at it at your leisure to kind of dissect it for yourself. And now what we have is a 1435 row data frame. That's all of the leaf nodes of the taxonomy. And if we look at the columns, we have everything labeled through cluster, group, subclass, class, and neighborhood. We've labeled every row along every level of the taxonomy. And so now we have all of the parent-child relationships in a single place, which allows us to do some interesting things. Let's say I wanted to know the names of all the classes in the taxonomy. I can just grab the class column and say, only give me the unique values, and those are the unique class names in this taxonomy. This is how you would have to do that before we had joined everything into that single table. You would have had to take the cluster annotation term data frame and filtered it down to the class level, ask for name, and again, ask for them to be unique. It's not a ton of code, but it is more code. Similarly, what we can do is we can limit ourselves to a single class and ask, okay, what groups are children of that class? Because remember, each row in this data frame now is a leaf level of the taxonomy, and it lists all of its parent levels. So just limiting down to this class and asking for the group names, we see these are the groups that are children of this class. So we can explore the taxonomy in a slightly more efficient way. Okay, so that's just constructing the taxonomy, but we haven't linked it to cells yet. So to join the taxonomy of the cells, we have to go to the cluster-to-cluster annotation membership file. Remember, that's where we link cluster annotation terms to cluster aliases. So let's download the cluster-to-cluster annotation membership data frame, like that. And there we go. And here we have, again, we have cluster annotation term labels, cluster annotation term set labels, and then cluster alias. This is that column I was telling you about that is ultimately going to allow us to link to cells. So this data frame, as you can see, it's big, 7,000 rows. That's because there's a row for every term in the taxonomy, and we really it's every row, every term in the there's a row for every term in the taxonomy and every cluster alias assigned to that term. We don't need all of that data. We just need the link between cluster alias and the leaf levels of the taxonomy, which we know from looking at the term set file is level 4. So we're just going to limit that data frame down to level 4. And now, again, our 1435 rows, one for each leaf node in the taxonomy. We only want cluster annotation term label and cluster label. So here we go. We only want cluster label and cluster alias. So we're going to do some filtering on the data frame. Again, we're going to limit down to cluster alias and cluster annotation term set label or term label. And then we'll rename cluster annotation term label to cluster label. Just standard pandas functionality to get us a 2-column database that has cluster alias and cluster label in it. Okay, so now we can go from taxonomy to cluster alias, but we still haven't gotten to cells. Cells are the cell linkage to cluster aliases in cell to cluster membership. So let's get that data frame. This is a much bigger data frame. It has one row for every cell. It has 1.9 million rows and 3 columns. It has the cell label. It has the cluster alias, so we can link each cell to the taxonomy. And then for convenience, because in this taxonomy, there's a one-to-one mapping between cluster alias and cluster label, it also has cluster label. This is not guaranteed. You should not count on this in future iterations of this data model. So we're gonna pretend that cluster label isn't there and just try to link cells to our taxonomy using cluster alias. So I'm gonna make our lives more difficult and only limit that data frame down to just cell label and cluster alias. And now we will link the cell to cluster data frame. We will join the cell to cluster data frame against the term to alias data frame earlier on cluster alias. And so that gives us now cell label, cluster alias, and cluster label, which we can now join to our taxonomy, because remember, our taxonomy has a cluster label column in it. So we can just do that join. And now we have a 1.9 million row data frame with each cell in it, and each cell is linked to every level in the taxonomy that cell is annotated with. We have now linked our cells to our cell type taxonomy. So that is how we represent cell type taxonomies in ABC Atlas Access. There are a few subtleties and a few caveats I would like to give you before we move on. The first is this is an evolving data model. We've been, ABC Atlas Access, I think we've released first data in it somewhere in 2023. It is now 2026. In that time, we've learned a few things about how we want to represent data, which means older taxonomies don't necessarily have all the files I was just telling you about. Let's look at the oldest taxonomy in ABC Atlas Access. Let's look at the whole mouse brain taxonomy. There are some quirks to the whole mouse brain taxonomy. The first quirk is how cells are linked to the taxonomy terms. So if we look at the whole mouse brain taxonomy and list all the files in it, you'll see there is no cell to cluster membership file. That's because the linkage between cells and cluster alias takes place actually in the cell metadata file associated with the single cell data itself. So I'm going to download that data frame now and load it. This is a big one. It's 4 million cells. So there. And you see this cell metadata data frame I've pared down to the columns I want, but you have cell label, and you have a cluster alias here. This is the cluster alias that links you to the taxonomy. It'll work the same way as the cluster alias we were working with in HMBABG, it's just in a different place. Similarly, if we look at there's another quirk about whole mouse brain that I'd just like to draw attention to. If we look at the cluster annotation term set, we see there are five cluster annotation term sets in this taxonomy. There's cluster, supertype, subclass, class, and neurotransmitter. I'm just drawing attention to that because neurotransmitter exists outside of the hierarchical taxonomy. It doesn't have any well-defined parent-child relationships. I can show you that by downloading cluster annotation term. Again, here we have our, it's about 7,000 rows, and it lists all the cluster annotation terms along with the parent terms and the parent term sets. And if I just loop through all of the term sets, subset the cluster annotation term data frame, and ask at each level in the taxonomy how many children call this level their parent, we see that nothing claims a neurotransmitter as a parent. Cluster, nothing claims cluster as a parent, but that's because it's the lowest level of the taxonomy. All of the clusters claim supertypes as the parents. All the supertypes claim subclasses as parents. All the subclasses claim classes as parents, but nothing claims neurotransmitter as a parent. It exists outside of the taxonomical hierarchy, which is something that you just have to discover. That's kind of the takeaway I wanted to give you with these subtleties is there's going to be a lot of working with this data that you're just going to have to discover. You have to explore the tables you've been given and see where the various columns you want. That's how you would discover the cluster aliases in cell metadata with the whole mouse brain. That's how you would discover that neurotransmitter exists outside of the parent-child hierarchy of the taxonomy. Hopefully, through this demo, I've given you some tools to do that exploration. Use Panda to slice up the CSVs, navigate through, figure out how columns are linked to each other, figure out how tables are linked to each other, and rebuild the data as you need it for your purposes. If you ever have any questions, of course, reach out over the Allen Community Forum. That's what it's there for. We are now going to work through a Jupyter notebook that shows how to work with H5AD files. H5AD is the file format that ABC Atlas Access uses to distribute the cell-by-gene data that comes with our data sets. It is built on top of the more common HDF5 format, which you may have heard of. The AD stands for ANData, and the AN stands for annotated. ANData is a Python library that is built around the idea of manipulating data and metadata in the same object in a Pythonic way. Their documentation is here at andata.readthedocs.io. As before, this notebook is in the GitHub repository that Rachel mentioned at the beginning of the webinar. It exists there in a much more verbose form. I've stripped out the extraneous text for the purposes of this demo, but one of the things the notebook does have is a link to the ANData documentation. So if you clone the repository, you will get links to the ANData repository. So with that out of the way, I'm going to do this in two parts. In the first part, I'm going to just talk about general considerations when working with H5AD files. This is not going to work with real data. I'm going to generate some cartoon data that looks real, and we're just going to play with it to see what kind of considerations you have to think about when manipulating H5AD files. In the second part, we will download some actual data from ABC Alice Access and try to manipulate it. So let's get our environment set up. The cells here are going to write out some cartoon data to disk. It's not real. It's got made-up numbers in it, but it looks real. And again, the point is to have something small that we can manipulate and in many different ways without dealing with the large data that comes from ABC Alice Access. I'm writing the data out in three ways. I'm writing it as a dense file, a sparse compressed column file, and a sparse compressed row file. Cell by gene data tends to have a lot of zeros in it, so you can save space on disk by saving it as a sparse matrix, which only stores the non-zero elements. For our purposes, there are two flavors of sparse matrix. There's sparse compressed column, which stores the elements in a way that it is easy to access them column by column, and sparse compressed row, which makes it easy to access in a row by row way. I'm also, as I said, storing it as a dense matrix where all of the zeros are stored on disk, and that file will be much bigger. And when I say much bigger, the dense file is 5 gigs. The sparse files are each about 2.5 gigs. But that is large enough to demonstrate the different performance considerations that come with writing the data in those two ways. Before we start manipulating this cartoon data that I just generated, let's talk a little bit about what is in an H5AD file. An H5AD file is a serialization of an AND data object, which is an object defined by the AND data library. AND data objects have a lot of attributes associated with them. For the purposes of ABC Alice Access, we only use 3. We use X, which is the array containing the actual cell by gene data. The AND data convention is that rows are cells and columns are genes. OBS is a pandas data frame. It is literally a pandas data frame. You can do anything with it that you can do with a pandas data frame that contains all the metadata for each row, each cell in the data set. And var is a pandas data frame with all of the metadata for each column or each gene in the data set. So let's load our dense H5AD file using the anddata.readH5AD function. Before I execute this, I just want to call attention to this configuration parameter, backed equals R. What I'm doing here is I am telling anddata not to load anything into memory until it is actually needed for a computation. For our purposes, that doesn't actually matter. These files are small, but there are files in ABC Atlas Access that are quite large. There's one in the whole human brain that is compressed down to 31 gigs. I think uncompressed it is 56 gigs. If you were to call read H5AD file without backed equals R, anddata would load all 56 gigs into memory at once, and depending on your computer, that might have consequences. So we call backed to tell anddata to leave the data on disk until the very last minute when I actually ask you to do something with it, which is just a good way to open any H5AD file whose size you are unsure of. So here, I've created my anddata object, and let's just look at those 3 attributes, X, OBS, and R. X, as you can see, it's an array, 300,000 cells by 5,000 genes. I guess I should scroll up really quick and show you that is in fact what I meant to write. 300,000 cells by 5,000 genes. But it's not an array yet. It's still on disk. We haven't loaded it into memory. OBS is a data frame with 300,000 rows. It is the metadata that we have decided to store for each cell in our cartoon data set. Here I just have 2 columns, the cell label and the donor label. And then var is a 5,000 row data frame, which has all of the metadata for the genes in our data set. In this case, just a gene ID and a gene symbol. If I want to get X into memory, I can call the toMemory method on it, and I will, because this is a small data set. So if I call toMemory and then look at X, you'll see something that actually looks like a NumPy array. There you have it. It's a NumPy array. I can do NumPy-like things with it. I can take the sum along each row. I can take the mean along each column. I can just manipulate it. I can do anything with it that you can do with any other NumPy array. That's all well and good. Now let's look at one of the compressed files. So get rid of that. Now let's load the CSR file, the compressed sparse row file. That is the sparse matrix that has been optimized for access along rows. Again, calling with backed equals R. Now we see that X is still on disk, but it is stored as a CSR dataset. It is not stored as a dense array. I'm going to load it straight into memory. And it's still a compressed sparse row matrix, but now it is in memory. And I can still do most of the operations I can with NumPy. I can still take that sum along rows, and I can still take the mean along columns. It just takes more time. Sparse matrices have a to-array function that returns them as a dense NumPy array, so I'm going to convert it into a dense array and show that those two operations are much faster on the dense matrix. On the dense matrix, both those operations take about half a second, more or less. On the sparse matrix, while the sum along rows is also about half a second, the mean along columns takes 1.5 seconds. It's 3 times slower because of how the data is stored and what has to be done to manipulate it. So that is just the tip of the iceberg in terms of what kinds of performance considerations you have to think about when you're loading an AND-data object or an H5AD file based on how the data has been stored. Now let's do a more detailed comparison. I'm going to open all 3 of my files, my dense file, my CSC file, the one that is optimized for column access, and my CSR file, the one that is optimized for row access. We can see they are all different. Let's say that out of my 300,000 cells, I only care about 6 of them. I care about the 0th, the 3rd, the 5th, the 290,000th, the 290,000th and 1st, and the 290,000th and 2nd. I can grab just those cells by subsetting the H5AD, the AND data object, like it were a NumPy array. I can just take the cell index array and pass it in like this was a NumPy array, and you see I get X is now 6 by 5,000. OBS now only has 6 rows, and var has the full 5,000 set of genes. The trick is each file is going to respond differently to this subsetting. So let's just compare all 3 files. You can see right there, you can just, without even seeing the numbers, that subsetting the compressed sparse column array by cells took noticeably longer than subsetting the compressed sparse row or the dense and data object by cells, even though the data returned by them is identical. So why is that? It's because of how the data is stored and how it has to be loaded into memory. I've written a utility function that you get with this notebook that will repeat the same operation we just did, but also keep track of how much memory is being used by the system as it is subsetting the AND data object. So let's run each of these AND data objects through that process and see not only how long it takes, but how much memory it requires. Again, you can tell that that subsetting the CSC file took a lot longer than subsetting the CSR file, or for that matter, the dense file. And now you can also see that the reason, or one of the reasons, is that the machine had to load the entire file into memory in order to slice the CSC file by cell. It did not have to do so for the CSR file or for the dense file. Conversely, if I were only interested in 6 genes out of my 5,000 genes, we would find the opposite. We would find that the CSC file is lightning fast, and the CSR file is very slow. So let's attempt that. So here's a gene index. We're going to just show that we can slice by genes in the same way that we can slice a NumPy array. See, I've got here's my subsetted var. A data frame only has 6 rows. OBS has all 300,000 cells, and X once again is 300,000 by 6. It just subsetted the genes as though this AND data object were a NumPy array. But now let's try to time that operation. We're going to try timing the from dense and from CSR and from CSC, and we see that again from CSR much slower than from CSC was or than from dense was, even though they return the same outputs. And again, the reason is the same. When you are subsetting a CSR file by columns, by genes, you have to load the entire file into memory. This is all well and good for our cartoon example, but as I said, there are some very large files in ABC Atlas Access, and so having to load all of them into memory just to get a subset of the genes could be very prohibitive, which is why it is important when you're working with an H5AD file to know how the data is stored in memory and plot your strategy for slicing and dicing the data accordingly so that you don't accidentally overwhelm the memory in your computer and cause the system to crash. Now let's apply what we have learned to actual data from ABC Atlas Access. Before we get to manipulating H5AD files and working with them, we have to think about how do we know which H5AD files we want to download. In many datasets in ABC Atlas Access, the cells are spread out amongst many different H5AD files. We did this to prevent anyone from having to download hundreds and hundreds of gigs of data unless that's actually what they wanted to do. A stellar example of this is the whole mouse brain dataset. I can show you by downloading the cell metadata table from whole mouse brain, and you can see what I'm talking about. The whole mouse brain cell metadata table has two columns, dataset label and feature matrix label. These correspond to the directory and file name kwargs of the ABC cache get file path function. These indicate which files each cell is stored in. So let's download one of these files. Let's download the one that corresponds to dataset label WMB 10xv2 and feature matrix label WMB 10xv2 thalamus. I'm picking it because it's small. It's only about 4 gigs. Here, I've downloaded it to my file, to my system, and it exists at this location now. Before we actually open the H5AD file, let's get the cell metadata for the cells we just downloaded. We can do that by taking the full cell metadata table and filtering it for dataset label is WMB 10xv2, feature matrix label is WMB 10xv2 thalamus. If I do that, I see that the 4 million cells in the original cell metadata table is now reduced to 130,055. Here's just the top 5 of them. Now let's read the H5AD file. We load the H5AD file into, or we open it. Again, backed equals R, so we're not loading it into memory. We see that var is a 32,285 row data frame with the metadata for the genes. It has gene identifiers and their gene symbols. And that obs is 131,212 rows long and contains the metadata for the cells in that H5AD file. This number is not the number we were expecting based on the cell metadata table. Recall, there were 130,555 cells in our pared down metadata table, and now there are 131,212 cells. What's going on? Well, the difference is quality control. Every cell that was sequenced appears in the H5AD files. Cells that fail quality control do not appear in the cell metadata table. That is one thing to be aware of when you're working with the data and metadata provided by ABC Atlas Access. Now, let's look at this detail at the data we've downloaded more closely. We see that examining X, this H5AD file is stored as a CSR dataset. The vast majority of datasets in ABC Atlas Access will be stored as CSR. A few are CSC, maybe one is dense, but CSR is generally the rule. And you have to consider that when you're manipulating the data. If we're only interested in a subset of cells, again, that's fine. Let's say we only wanted these 6 cells. We could just, subset quickly, as though this were a NumPy array. You see obs is down to the 6 cells we selected. X has only 6 rows in it. And we can even convert it into a NumPy array. No real consequences to downloading or to downsampling by cells because the data is stored as a CSR matrix. But what if we want to downsample by genes? What if we're only interested in a subset of genes? Here, I'm going to randomly pick 200 genes to pretend that these are my genes of interest. As we saw in our theoretical consideration of H5AD files, if we were to try to downsample a CSR matrix by genes, the ANData would load the entire file into memory. For this file, that doesn't have many consequences. The file is only 4 gigs in size, but there are files that are tens of gigs in size. Many tens of gigs of size, and they could quickly overwhelm your system. There are many ways around this problem. The way I'm going to recommend it is that we assemble our subset of genes gradually, chunk by chunk, in cell space. An ANData object provides a function, chunkedX, that gives you an iterator that iterates over chunks of cells. You just tell it how many cells at a time you want. Let's say we wanted 10,000 cells at a time. We could instantiate a chunk iterator using this function call, and then to see what the chunks look like, I'm just going to call next on my row iterator, and you see that the chunk we get back is a 3 tuple. The first element in the tuple is the CSR representation of the chunk of cells we got, and then we have the indices of the first the indices indicating which cells are in that chunk. We can use this row iterator to gradually build up our subset of genes. ANData also has a function concat, which allows you to take a list of ANData objects and concatenate them into one big ANData object. So what I'm going to propose we do is we create a chunk iterator that iterates over our data set 1,000 cells at a time. It's going to go over each of those chunks. It's going to create a new ANData object for each 1,000 cell chunk, downsampling that, downsampled by genes to only have the 200 genes we care about. And then we're going to build up this list of small ANData objects, which we will then use concat to build into one large ANData object. So here, I've written code to do that here. Let's just run it. It's not the fastest thing in the world, but there. It took about 7 seconds. And we see we got back our TH gene subset, which should have 200 rows. We have our 200 genes of interest and all of the 131,000 cells in it. Now, we could have just done that by treating the ANData object as a NumPy array and just slicing directly on the genes. And if we run that here, we'll find that actually in this case it's a little faster. It only took 2 seconds here, but the difference again is the memory footprint. Here I'm just proving that the outputs of the two processes were the same. If I use the read and profile functions that I wrote again to perform these operations, but also keep track of how much memory is used, what we will see is that, again, if you are directly slicing the H5AD file on genes, and it is a CSR H5AD file, you have to load the entire file into memory. No consequences for a 4 gigabyte file, but once you have a 30 gigabyte file or a 50 gigabyte file, you might not want to load the whole thing into memory. Which is why I suggest when you are slicing in H5AD data along cells, genes, you keep track of the shape of the data as it is stored and devise a strategy so that you're never loading too much data into memory. That is the price we pay for storing our data as sparse data that doesn't eat up all of our hard drives. So now let's simulate an actual scientific use case. We are going to find some interesting data, download it with ABC Atlas Access, and then attempt to find marker genes between some of the cell type populations in the cell type taxonomy. So let's imagine that we were tooling around in ABC Atlas. Here I am at brainmap.org. I can go to cell type taxonomies. I will go to Allen Brain Cell Atlas. And let's say we're interested in the whole mouse brain. We can just click here and get a visualization of the whole mouse brain data. I actually get 2 visualizations. We have the spatial transcriptomics on the left and the single cell transcriptomics on the right. I'm just going to worry about single cell data right now, so I will delete this visualization. Let's say I was interested in how the cells are characterized based on the neurotransmitters they respond to. And let's say I knew that I wanted to look at serotonergic cells. So I'm just going to click right here under neurotransmitter type, and only the cells marked as being serotonergic will now show up in my visualization as colored. Now the first thing you notice in the UMAP is the serotonergic cells seem to be widely distributed in UMAP space. There's a big cluster of them here, but then there's a satellite here and another satellite way over here. And then if I color the cells by their supertype, so I go over here to 10x whole mouse brain taxonomy, click on the little color drop, I select supertype, I can see that in fact those islands are distinct supertypes. And here we have a few supertypes hanging out in the middle there. So what gives? What's going on here? Why are these supertypes different from all the other serotonergic supertypes? Well, ABC Atlas Access will help you get the data to help you answer that question. So let's do that. Again, here is a notebook I put together for this use case. It is available in the GitHub repository that Rachel mentioned at the top of the webinar. Let's just get ourselves set up as per usual. So the first thing we have to do is figure out which cells are serotonergic. So that's going to involve downloading the cell metadata table from Whole Mouse Brain 10x. We're then also going to need to annotate that table with the neurotransmitter annotations and the supertype annotations, so we can figure out which cells are serotonergic, and within the serotonergic cells, which cells belong to which supertype. So this is going to very much mimic the work we did in the cell type taxonomies notebook. I'm going to download the cell metadata table. Now I'm going to want just look at the cluster annotation term set table to see what the labels are associated with neurotransmitter type and supertype. And then I want that cluster to cluster annotation membership table. That's going to be the table that associates clusters and cluster aliases with neurotransmitters, classes, subclasses, supertypes, etc. So download that. Now I'm going to subset the cluster to cluster annotation membership table to only include annotations related to neurotransmitter type. I'll do some clever renaming of columns so they're a little more neurotransmitter centric. Keep only the columns I want. And now, boom, I have a data frame that has cluster alias and it's all and then all the neurotransmitter types associated with those cluster aliases. Remember, the cluster alias is what is ultimately assigned to the cells in the whole mouse brain data set. Now let's do the same thing with supertypes. Take the cluster to cluster annotation membership data frame and only select for cases where cluster annotation term set label is the supertype. Rename those columns so that they're supertype specific. Down select to the columns we want. And now we have a data frame linking cluster alias to supertypes. So now I can take those two data frames, the supertype data frame and the neurotransmitter data frame, and join them to the cell metadata frame on cluster alias, which I will do here. So just taking the whole mouse cell metadata and joining neurotransmitter DF and supertype DF on cluster alias, which means we now have a 4 million row data frame listing every cell in the whole mouse brain data set, and additionally its neurotransmitter annotation and its supertype annotation. So now we have the metadata, but we need to get the actual data. And remember, we're only interested in serotonergic cells. So let's downsample our metadata table to only contain cells that have been annotated with the neurotransmitter sero. And here our 4 million row data frame is now a 1469 row data frame, much more tractable. But again, it has everything we need: cell label, dataset label, feature matrix label, neurotransmitter, and supertype. So that's the metadata. Now let's get the data. Recall from our H5AD tutorial that the columns in the metadata table that associate a cell with an H5AD file are the dataset label and the feature matrix label. So let's take our downsampled serotonergic data frame and limit it to only the dataset label and the feature matrix label, and then we're creating a new data frame here, and we're just going to drop all duplicate rows. The effect of this is we're just going to list all of the unique combinations of dataset label and feature matrix label in our metadata data frame. And so you see we need to download 3 files. They're all from the 10xv3 platform, and these are the 3 files we need to download, which we will do here. This cell is actually going to take you about 20 minutes to run. It will download 25 gigs of data. I have already done that, so I can just run it with impunity, but just be aware that is the resource cost of doing this. There's smaller data sets in ABC Atlas Access, but there's no such thing as small data in ABC Atlas Access. Additionally, let's just download the data frame that contains all of the metadata about the cells in the whole mouse brain 10x data set. So that's this gene table here. I could show you what is in that data frame very quickly. It's kind of an expanded version of var. It has the gene identifier, the gene symbols, also more common names for the genes, any NCBID identifiers that we've been able to associate with these ensemble IDs, and any comments that our scientists annotated the gene with in the course of assembling the data set. Okay. So we now have a metadata table telling us where all the serotonergic cells are. We have downloaded all the H5AD files containing those cells. Now what we want to do is create an ANData object that contains just the serotonergic cells, that contains the cell by gene expression data, an OBS data frame for the serotonergic cells, and a VAR data frame for the full gene panel of the 10x data. That's what the next cell is going to accomplish. I'll kind of walk through the parts here. First, we're going to create an empty, a NumPy array of zeros that is just number of cells by number of genes long. Since we're only dealing with 1,500 cells here, we can afford to hold all of this in memory. We don't need to do anything clever about writing to disk. This is just a dict that is going to tell us where in which row in this expression data corresponds to which cell. That is going to be governed by the order of the rows in our serotonergic metadata table, because we're going to use that as OBS in our AND-data object. After that, we will just loop over every file that we downloaded, every dataset label, feature matrix label combination in our files needed data frame. And we will get the path to the file, we will load that file, and then we will subset that file, use it for only the cells in that loaded file that are in our serotonergic data frame. And then we will assign the expression values for that subset of cells to the appropriate rows of our empty expression matrix data. Then what we will have is we'll have our serotonergic metadata table, which is going to serve as OBS. We will have our gene metadata table, which serves as VAR, and we will have that expression data NumPy array, which is going to be X. And we can use all of that to instantiate a new AND-data object. And we will then have this sero AND-data object, which is an AND-data object containing the data and metadata for just the serotonergic cells in the whole mouse brain data set. So let's run this cell. I believe that we'll have completed. See, nothing doing. So now what we can look at what we have created, and we can see the OBS for this sero and data is in fact our metadata table of serotonergic cells, and var is the full 32,000 row data frame of the metadata for the genes, and the X is 1469 by 32,000 array of gene expression data. Excuse me. So now what we want to do is we want to take the various, remember, because the visualization we're working off of is this. We want to figure out why this supertype and this supertype are so unlike these supertypes in the UMAP, despite all of these being serotonergic cells. So what we're going to want to do is take our sero and data object and subdivide it by those supertype assignments, which we can do because there is a supertype column in our OBS data frame. C'est voila. So first, one of the supertypes we are interested is the 0893 MBMY TPH2 GLUT06 supertype. So we can limit ourselves to just cells that have been annotated with that supertype. Similarly, we are interested in 0894 MBMY TPH2 GLUT07 supertype, so we can limit ourselves to create an AND-data object that is just those cells. And then finally, we can create an AND-data object that is every cell that is not either supertype 0893 or supertype 0894. And what we have just done in these cells is we've created Boolean masks that are true for the cells we want and false for the cells we don't, and just use those to slice the AND-data objects and create other subsetted AND-data objects. And we can see now that we have subdivided our sero AND-data object accordingly. So let's find genes that distinguish supertype 0893 MBMY TPH2 GLUT06 from this other supertype AND-data object. So we're just going to not put too much thought in this. We will use the SciPy stats library, and we'll do a Wilcoxon rank sum comparing the expression matrix of supertype 0893 with the with the expression matrix of what we call the other supertypes. Similarly, we will let's just take the mean expression of every gene in the two populations, one for 0893 and one for the other supertypes, and just calculate the difference in the two mean expression values. Now, without thinking too hard about it, I'm going to claim that a good marker gene is anything that has a Wilcoxon rank sum p-value less than 0.01 and has at least a difference in log2 normalized expression of 5. And so I'm just going to create a Boolean mask that only selects cells that match both of those criteria. And we see there are 23 of them out of our 32,000 genes. So I'm going to just subset our delta array and gene symbols to only have those 23 valid marker genes. And then I want to find the one that has the maximum difference in expression between supertype of interest and the other supertypes. Turns out that gene is ZEB2. We can do a quick plot to see if I believe that. So import matplotlib. Figure out which column in our ANData objects corresponds to ZEB2. And then we're just going to loop over the two populations. We're going to loop over let me generate the figure. We're going to loop over every supertype in the serotonergic cells and just plot a quick histogram of distribution of ZEB2. And what you see is not a lot of ZEB2 going on until you get to our two interesting supertypes, 0893 and 0894. If I go back to ABC Atlas, here's my visualization. I'm going to click over here, duplicate visualization, so I have side-by-side view of the same thing. I'm going to sync the two cameras so I can now move the two visualizations in sync. And I'm going to click over here. And I'm going to color this. So this visualization is colored by supertype on the left. I'm going to color the visualization on the right by gene expression. Go to the little gene-looking thing. Get rid of that, and I'll type in. Let's look at it by ZEB2. See, here's the full histogram ZEB2 distribution. The distribution of ZEB2 expression in the cells we filtered by. And now I'll click the color drop to color the right panel by ZEB2 expression. And you can see here, 0893, lots of ZEB2 expression. 0894, lots of ZEB2 expression. As you see, as those cells highlight on the right, they're very purple. And then everything in this middle blob, there's some ZEB2 there. If I were to filter on ZEB2 expression, here, I can just say, okay, I only want the most expressing cells in that left panel. Come on. You can see this middle blob basically vanishes, but we still have our two interesting island supertypes. So we have successfully, I would argue, found a marker gene for those two supertypes. This is the kind of analysis that we hope to enable with ABC Atlas Access. This is why we want to give everyone access to the data behind ABC Atlas, so you can do these kind of deep dives and help further your analysis of this massive data set. Thank you for your attention. Again, reach out over the Allen Community Forum if any of this raises questions in your head, or if we can help you with anything else. Thank you.
Here are some useful links and resources, including the link to the community forum, which Chris and Scott both mentioned. community.brain-map.org. You can create an account, ask any questions, see questions other people have asked, and you can talk with both Scott and Chris and myself directly through the community forum. You can also continue watching this webinar series and register for the live webinars at alleninstitute.org/events, or you can watch them recorded here on YouTube. Lastly, we would also like to thank our funding sources, and thank you for joining us at this webinar.